Oxidative phosphorylation, formation of ATP. My name is Dr. Alamzev. I am a professor at the Department of Biochemistry, University of Malacca. There are some questions that we need to answer today. The first one is somehow they are similar. How FADH2 and NADH2 are consumed in the mitochondria. And the next question would be how oxidation of NADH or FADH are coupled to the phosphorylation of ADP. So we are going to answer these questions. NADH and FADH are present in mitochondria. We will be talking about these two which are present in mitochondria, not the one which are present in the cytosols, because we will discuss them later on. There are two important mechanistic theories. The first one is chemical coupling theory, and the second one is chemi-osmotic theory. So we will start with the first one, chemical coupling. It was widely believed that ATP synthesis occurs by chemical coupling between 1940 to 1960. High energy intermediates similar to 1,3-bisphosphoglycerate, which is used for substrate level phosphorylation, was once proposed to be produced first from the electron flows on both the mitochondrial and chloroplast membranes. Phosphorylation or phosphorylated proteins, the intermediates, is found in the action of phosphoglycerate mutase enzyme and phosphoglucomutase enzyme were also hypothesized, but neither were ever revealed despite intense effort by many investigators over several decades. This chemical coupling is similar to substrate level phosphorylation, in which ADP combined with the PI in the presence of energy, which could be the heat, and this form ATP. The second theory is chemi-osmotic theory. This theory was proposed by Peter Mitchell in 1961 to explain the coupling of the electron flow and ATP synthesis. This is his paper, which was titled is A Paradigm Shift in Understanding Bioenergetics. This was published in 1961 in Nature. And this tells us the whole theory. So the term chemi-osmotic emphasizes that chemical reaction can be coupled to osmotic gradient in the mitochondria. You must remember that proton gradient across the membrane in the mitochondria is responsible for the formation of ATP. This is the main point. So here you can see that when NADH gives its electron to the complex 1, here four protons are pumped to the inner membrane space, and then these electron and proton from the complex 1 are transferred to coenzyme Q, and then in the complex 3, when it passes through the complex 3, four protons are further transferred to mitochondrial inner membrane space. Similarly, when electrons are transferred to cytochrome C and then cytochrome 4, there are two protons are pumped to the inner membrane space from the matrix. In this time, electron water molecules are formed. So the electron flow and ATP synthesis are separate event coupled through a transmembrane gradient. So when protons are pumped to the membrane space, they are pumped back to the matrix and ATPs are formed. Formation of ATP through oxidative phosphorylation is carried out by enzyme called ATP synthase. This ATP synthase is present in the inner mitochondrial membrane. The structure consists of F0 and F1. F0 is the part which is embedded in the membrane and which has a rotor which is called C, and there are some thread in the head part, and here when the electrons are pumped from the matrix from the inner membrane space back to the matrix, this B alpha, this part of the head, the F1 part, they rotates, and the energy at this point is sufficient to combine ADP to PI to form an ATP. So we need to know that this enzyme is made of two distinct components. F1 are peripheral membrane proteins, and also F0, which is integral to membrane, specifically the inner membrane of the mitochondria. The structure of this enzyme was discovered by Raker and colleagues in 1960, for which they were awarded, Raker was awarded a Nobel Prize in 1978. ATP synthase is a splendid molecule, and it comprises a proton channel F0 and an ATPase F1. The rotor, the C-ring, and the stack, the starter, and the remainder. So this is the whole composition of the ATP synthase. Here you can see that this pathway ATP formation is coupled to electron transport chain. So here we can say that this coupled pathway is consists of two parts. The first coupling, which consists of the whole electron transport chain system, while the second coupling is the proton coupling, or the second coupling, where the protons are transferred back to the matrix. So here this chemiosmotic theory have three main postulates: an electron transport chain providing the energy per proton transfer from one side to other side of the membrane, and the second one is ATP synthase, synthesizing or hydrolyzing ATP through proton translocation, and the third one is impermeability of the inner mitochondrial membrane to ionic species, thereby producing including protons. So these are the main three postulates of the Mitchell theory. The basic requirements for oxidative phosphorylation is a coupling between redox processes, proton translocation, and ATP synthesis. Such global coupling can arbitrarily be divided into two distinct phases. Number one, a coupling between the oxidation reductive process and the protonic translocation, referred to as redox coupling. This is the or the first coupling, which is we call the electron transport chain. So this is the first part, which we call redox coupling or the first coupling. The second is the coupling between proton accumulated on the P side of the membrane moving to the N side through the ATP synthase, which determines the synthesis of ATP. Also, we call this coupling as a proton coupling or second coupling. So in this case, you should remember that these processes are coupled to each other. But experiments demonstrated that proton translocation is crucial step for the proton coupling between protonic moment and ATP synthesis. There are a lot of new experiments on this subject. The recent study updated our understanding of the oxidative phosphorylation. However, there are still several controversies to prove right or wrong on the basis of experimental proofs. For example, water molecule is considered to form hydronium ion occurring fastly than simple proton release to the aqueous medium. So it is another experiment. A primary role for membrane buffering and proton mobility in general can be hypothesized. Also, results suggested that proton can efficiently diffuse along the membrane surface between a source and a sink. For example, the proton ATP synthase without dissipation losses into the aqueous bulk. So these were the results, including these one. The hydronium ion can be transported through HV1 channel. In 2006, a direct proton transfer was also proposed to couple the respiratory pathway of complex 1, 3, 4 with ATP synthase. Another one says the exergonic process of proton salvation would release energy. So when proton is again solved, it will produce energy. The existence of protonic current internal to the membrane with the formation of possible circuits traveled by positive elementary charge, thus realizing a localized coupling that excludes an osmotic nature of the process. So in the summary, you can see here we have three main processes in electron transport, a hydrogen movement, and ATP formation. So in the complex one, the NADH gives electron to the complex one, where the protons are transferred to the inner membrane space. Similarly in complex 3 and complex 4. While when these protons are coupled back, they are coupled to the ATP, the transfer of proton back to the matrix using ATP synthase. And here this energy is sufficient to produce, to combine one ADP to PI to form ATP. So here you can see there are two types of coupling. One is the electron transport coupling and phosphorylation.